Welcome back students. Today we are going to discuss unit 5 and in this particular unit we are going to discuss plant growth and development part 1. Before I start the main content, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT, have any concern, any issue, please contact us at sathee@iitk.ac.in and we shall remove the images. So in this particular video lecture, we are going to discuss these points: plant hormone, growth regulators, action of plant hormone in related to seed dormancy, germination, apical dormancy, senescence and abscission, application of synthetic growth regulator, and a brief account of growth and movement in plants. So what is growth? First, we have to understand the term growth. Growth is a fundamental and noticeable character of living organism or any living being. So it defined as a irreversible permanent increase in the size of an organ or its part or in a cell. Growth is irreversible and it's a permanent change, right? Permanent increase in size of an organ, its part or in a cell. Growth involve various metabolic process in the form of anabolic form or catabolic form that consume energy, right? So leaf expansion. Expansion of a leaf is a example of growth. So growth is measurable. If we are discussing it about at cellular level, that time we can say if protoplasm increase, that time we can say cell increase in volume, okay, or there is a growth in a cell. So at cellular level, it results from an increase in protoplasm. Protoplasm increase is difficult measure directly, so growth is measured by proportional quantity, right? So growth is measured by parameters like fresh weight, dry weight, length of the plant, what is the area, volume, cell number. So these are certain parameters uh, with which one can analyze the growth fact, okay, of a plant. So maize root apical meristem can produce over 17,500 new cells per hour. Per hour, right? A maize root apical meristem. Similarly, watermelon. In this watermelon, cells can be increased in size by up to 350,000 times. Growth can be expressed as an increase in cell number or cell size. Clear? So, and for if we are talking about the pollen tube, so for pollen tube, growth is measured by its length. For leaf, dorsiventral leaf growth is measured by the surface area increase. Clear? So conditions for growth. Conditions for growth. What are those essential conditions? Essential conditions for growth include water, oxygen and nutrients. Water, it is necessary for cell enlargement, its turgidity and adding extension growth. Water provide a medium for enzymatic activities too. Oxygen, it release as a metabolic energy needed for growth. So what are those essential conditions? Number 1, water, number 2, oxygen, and number 3, nutrients. For nutrients, as a macronutrient form and micronutrient, both are required for protoplasm synthesis and its energy. Temperature, optimum temperature range is crucial. If there is any kind of deviation in temperature, it can be harmful for the plant growth. Environmental signals like light, like gravity, it also affect certain growth phases of the plant. Okay, now plant growth regulator, PGRs, plant growth regulators, it also referred as plant hormones or phytohormones. It is a characteristic of plant growth regulators. Like, what is the composition? Plant growth regulators are small in size. These are simple molecules with diverse chemical composition. Like indole some indole compounds like indole acetic acid, IAA, like some adenine derivatives like chitin, it is a adenine derivative. Certain kind of carotenoid derivatives also act as a growth hormone like ABA is an example for this carotenoid derivative. Some like gibberellin, GA3, it is these are terpenes. Some gases are also act as a growth hormone like ethylene. Okay, so these are small molecules, simple molecules with diverse chemical composition. We have to learn this thing. What are the examples for indole compound, example for adenine derivative, examples for gases who are act as a growth regulators. Now, growth promoters. Out of these, some hormones act as a promoter, some act as a inhibitor. So, all those hormones, PGRs, growth regulatory hormones, which involve in some kind of activity like cell division, like cell enlargement, pattern formation, tropic growth, flowering, fruiting, and seed formation, those are comes under growth promoters category, like auxin, like gibberellin and cytokinin. Auxin, gibberellin, cytokinin act as a growth promoter. They all are involved in cell division, cell enlargement, tropic growth, flowering, fruiting, and seed formation. Some inhibitors, those are involved in abiotic or biotic stress conditions. Those if plant respond to a wound, for biotic response, a biotic or abiotic stress condition, those are comes under growth inhibitor category. So these growth hormones involved in the dormancy phase and or we can say abscission, like abscisic acid, like ethylene. But ethylene can fit both group. So this one is important. Sometime it act as a inhibitor, sometime it act as a promoter. But mostly, primarily we can say ethylene can fit in under growth inhibitor category. Clear? Now, discovery of plant growth regulators. Discovery of 5 plant growth regulator was accidental. Charles Darwin, Charles and Francis Darwin observed canary grass. What type of grass? Canary grass, coleoptile, growing towards the light, which is known as phototropism. And then they conclude that coleoptile tip transmit a influence causing blending. Okay. Similarly, this is related to Darwin, French Darwin, and another one, F.W. Wendt, another scientist, he isolated auxins from oat seedling coleoptile tip also. Okay, so this is how accidentally some scientists observe the role of plant growth regulators, and they further isolate these plant growth regulators too. Gibberellic acid was isolated by Kurosawa in 1920-60, and they linked the symptom of Bakani disease in rice, which is a disease of rice, Bakani disease in rice, to sterile filters of Gibberella fusicori fungus. Right? So, active substance, this is related to this kind of substance which is identified as a gibberellic acid. Similarly, this chitin observed by F. Skoog and his team, he observed callus formation, callus proliferation in tobacco stem requiring auxin and supplements. Similarly, Miller also worked on this plant growth regulator related work. He crystallized and named the cytokinin promoting substance chitin. So he F. Skoog just observed while Miller isolated or crystalline this chitin. Abscisic acid. In mid 1963, independent researcher identify abscisic acid as a inhibitor B, abscission 2 and dormin. Later found chemically they are identical, and then they named it as a abscisic acid. Ethylene. For ethylene, this was observed by H.S. Cousin, and he found this ethylene as a volatile substance from ripe orange which accelerate banana ripening, and he identified later it as a ethylene which is a gaseous PGR, plant growth regulator. Now auxins from auxine means to grow. It is isolated from the human urine. It also includes the natural compounds like indole 3 acetic acid, IAA, indole 3 acetic acid, and synthetic like NAA and 2,4-D. IAA, NAA, and 2,4-D. They all are belong to auxins category. These auxins are produced by stem and root apex. They migrate to the and produced at the tip and then migrate to the target region where there is a requirement for this particular auxin. Okay, so it promote, what is the role of auxin? It actually promote rooting in stem cutting. Some plants we used to grow with the cuttings like rose, right? So auxins promote rooting in stem cutting and flowering like pineapples. Okay, so with the help of this picture you can easily identify or analyze that this auxin diffuse to the shaded site of the plant causing cell elongation and show positive phototropism. Okay, now auxin prevent early stage fruit and leaf drop, early stage fruit and leaf drop, but it promote abscission of mature one. Okay, control apical dominancy. Auxin control apical dominancy, shoot tip removal. So shoot tip removal stimulate lateral bud growth. Okay, if we remove this shoot tip, then it will promote lateral bud growth. A, apical dominancy in plant. Normal, it is normal. A plant with apical bud interact, a plant with apical bud removal in picture B. So growth of lateral bud occur in the second plant after the removal of the tip. Clear? So it induce parthenocarpy also, in which grow in tomato, and it also serve as a herbicide. So auxin also induce parthenocarpy, seedless fruit. Okay. 2,4-D, it is a type of auxin. It target dicot weed without affecting mature monocot plant. So 2,4-D also act as a herbicide because it target weedicide, not herbicide, weedicide. It target dicot weeds without affecting mature monocot plant, and sometimes question asked on this line. It also influence xylem differentiation and seed and cell division also. Okay. Now zebralin. Zebralin, it is a type of plant growth regulator, exists in over 100 variant. Okay, found in fungi and higher plants as a GA1, GA2, GA3 and so on. Zebralic acid GA3 was first discovered and remained extensively studied. Which one? GA3. This was the first discovered. Sometime question also asked on this particular line. So all gibberellins are acidic in nature. They induce various physiological response in plants. They promote axis elongation, use lengthening of the grape stalk, and improve apple shape while delaying senescence. Means this elongation of the plant axis growth, distance between the two nodules like this. And gibberellin actually enlarge the size of the fruit, right? For apple, for grape, and delaying senescence. GA3 accelerate the melting process in brewing. Okay, malting process in brewing, and boost sugar cane yield by increased stem length. Boost sugar cane yield by increase its stem length. These are again important line. So if we spray this gibberellic acid GA3 on juvenile conifers, it will speed up the maturity for early seed production. Okay, early seed production. Gibberellin induce bolting in plants like beet and cabbage. It will promoting internode elongation, as you can see over here in this picture. Okay, just before the flowering. Okay. Now cytokinin. Cytokinin initially discovered as kinetin from autoclaved herring sperm DNA specifically affect cytokinesis. Kinetin. It is a modified form of adenine. We have discussed this in 2 or 3 slides before. It is a naturally occurring hormone. Okay. Gelatin. This gelatin isolated from the corn kernel and coconut milk, and it possesses cytokine-like activity. Clear? So natural cytokinin, these are synthesized in the region with rapid cell division. Like, what are those locations? Root apical region, shoot buds, these are also having rapid cell divisions, and young fruits. So these are synthesized in the region where rapid cell division is the main task. They facilitate new leaf chloroform for production, lateral shoot growth, and adventitious shoot formation. They overcome apical dominancy. Cytokinin also promote nutrient mobilization, delaying leaf senescence. Ethylene. Ethylene, it is a gaseous hormone. We have discussed it, right? And these are used in fruit ripening. These are mainly involved in fruit ripening. It promotes horizontal growth of the fruit, axis swelling, and apical hook formation in dicot seedling. Ethylene also triggers senescence and organs abscission, especially in leaves and flower. Crucial, this ethylene are crucial for fruit ripening by enhancing respiration rate. Which is known as respiratory climacteric. Clear? Ethylene also initiate germination, elongation, it elongate internode and petiole in deep water rice plants. And it stimulate root growth. Ethylene induce flowering. It also synchronize fruit set in pineapple and mango. Ethaphone. Okay, ethaphone, it is a widely used source of ethylene, hasten fruit ripening, accelerate accession, and promote female flower in cucumber. Which one? Ethaphone. It is a type of ethylene which is widely used by the people. Abscisic acid, ABA, which is known as ABA. ABA was initially discovered for its involvement in regulating abscission or dormancy. It acts as a plant growth inhibitor. So it inhibits seed germination. It stimulates stomata closing, means reduce transpiration. It involved indirectly to reduce transpiration. Okay, so it also stomata closing, so reduce water loss. It promote fruit ripening. It increase shelf life of the plant, which and tolerance of the plant for transplanting seedling. Seed treatment for hybrid seed production. It promote cold hardiness, and ABA comes under plant growth inhibitor category. It enhance plant tolerance to various kind of stress. So it is also known as stress hormone. as it induces dormancy. It allows seed to withstand under unfavorable condition. Under unfavorable condition. It this ABA is important for seed development and maturation. Often act as an antagonistic to gibberellins. So just what kind of work it is? Just opposite type of work of to the gibberellin. Gibberellin promote growth, development. It just um, induce dormancy. Okay, and work under stress condition. Okay, other factors like different PGRs means plant growth regulators have complementary or antagonistic role during plant growth development. Complementary or antagonistic role during plant growth and development. PGR along with genomic control and extrinsic factor like temperature and light, they influence plant growth and development. Dormancy in seed or buds, abscission, senescence and apical dominance. Extrinsic factor play a role in events like vernalization, flowering and plant movement. So this is all about the today's video lecture which is related to growth hormones. I hope most of the things has been cleared. Still you have any doubt, any question related to this, please let me know. We will discuss it later on. Till then, take care, bye bye and keep learning. Namaskar.